In the last lecture, we were looking at the structure and function of carbohydrates. Today, we will complete that, the structure and function of carbohydrates. And then the next time we meet, we then take the metabolism of carbohydrates. So today's lecture shouldn't take so long, maybe about 45 minutes, maximum 1 hour. We should finish dealing with the rest of the things under the structure and function of carbohydrates. So that's the next time we will go into the metabolism. Disaccharides. I mean, we were looking at just the monosaccharides and then the modifications of the monosaccharides. And then we got to this and we decided that let's leave it because from here we start to see the monosaccharides now joining to form bigger molecules, and starting from the disaccharides where we see the disaccharides of one glucose molecule and one fructose molecule, maltose, two glucose molecules, and lactose consisting of glucose and galactose. So let's look at the structures of these disaccharides. We take the first one, which is sucrose. That's the table sugar. And so we see the monomers, glucose, which is at the top, and we see fructose, which is below. And the two of them form a glycosidic bond to give it a sucrose molecule. Now, I want you to pay attention to this structure. Usually, you've been seeing, say, maybe more disaccharides or carbohydrate molecules with glycosidic bonds, which are formed between the carbon number 1, and if it was another glucose molecule, it would be carbon number 4, which is usually the 1,4 glycosidic bond that you refer to. But this particular one that you are seeing, you see the bond is between the glucose molecule, carbon number 1, and then the fructose molecule, carbon number 2. That's what you are seeing there. And you see you have this bond being an alpha 1 beta 2 glycosidic bond. What does it mean? If you take a glucose molecule, you see the glucose molecule is in its alpha configuration. So you see glucose molecule, the hydroxyl group on the anomeric carbon is pointing downwards. So that is an alpha configuration of the glucose molecule. Now when you take the fructose molecule, you see the hydroxyl group is facing upwards. That is the beta configuration. And it's even written below. You see alpha D glucose and beta D fructose. And so you have this bond, alpha 1, beta 2 glycosidic bond or glycosidic linkage. You see when these bonds are formed, you see there's some water molecule that is given out there. So that is a form of a condensation reaction. It's a removal of water molecule from that process there. Now the sucrose, when you look at it, you realize that we refer to sucrose as a non-reducing sugar, and mean, that is because you see that the carbons that are reactive, both of them are involved in the bond, the bonding, the glycosidic bond. Carbon number 2 and the carbon number 1 of the glucose molecule, carbon number 2 from the fructose molecule. And so you see the ends, the glucose molecule, carbon number 4, that is its non-reducing end. Similarly, the carbon number 5 here is also the non-reducing end of the sucrose molecule. But of course, we are not going into that, reducing and non-reducing sugars, just to mention it. Perhaps when you were in first year, when you were doing carbohydrates, because I remember you have SCT 106, some aspect of carbohydrates metabolism, and you do some carbohydrate structure there as well. So you would have met something of the sort. Maltose, which is consisting of 2 glucose molecules. So now you see the 2 glucose molecules, both of them are in alpha configuration. The hydroxyl group of the hydroxyl group, which is on the anomeric carbon of the 2 glucose molecules, are pointing downwards. But that's not even much of an issue per se, but you see this hydroxyl group, which is on carbon number 1 for this glucose molecule, gives out a hydrogen atom, and this hydroxyl group rather gives out a hydroxyl group, and that will result into the formation of water. So that's another condensation reaction. I believe when you were doing carbohydrates in first year, you would have seen the formation of hemiketals and hemiacetals, and then that's further. So the formation, I think if you remember the formation of the ring structures in first year, and those structures, if it was the aldoses, then the structure that was being formed was a hemiacetal. But if it was a ketose, then it was a hemiketal. So that when you see the hydroxyl group that is on the anomeric carbon coming to react, it is reacting as a hemiacetal, whilst that hydroxyl group on carbon number 4 of the second glucose molecule is acting as an alcohol. So you see that reaction, it may look to you as if maybe some acid-base reaction because there's some water formation there, but that's not what it is. And you see the glycosidic bond is formed between carbon number 1 and carbon number 4. And so that bond is referred to as a 1,4 glycosidic bond. So that's an alpha 1,4 glycosidic bond formed between 2 glucose molecules to give you the disaccharide alpha maltose. Now we move to the last one, which is lactose. And lactose is made up of glucose and galactose. Glucose and galactose. So if you look at it as the reverse of the reaction there, because here it's showing, say, the breakdown. But if you look at it as a reverse, so that you see those molecules, but actually, usually you should be having a glucose and a galactose that will be bonding to form the glucose, to form the lactose molecule. And here you see the bond which is forming between. So if you pay attention, you see this is a galactose molecule, this is a glucose molecule, And I'm saying that the reaction is showing, say, the breakdown of lactose. But if you look at it as a reverse, it will be the formation of the lactose molecule. So that you have a galactose molecule and a glucose molecule that will bind together. But there's something interesting also here. When we refer to the alpha and beta configuration, we talk of the fact that if the hydroxyl group is facing downwards, that is an alpha configuration. And if it's facing upwards, that is a beta configuration. But here you are seeing something else, where you see the hydroxyl group written, by a horizontal bond, and there is an asterisk on it to give you a note to tell you that if you are drawing a structure and you don't want to show whether it's an alpha or beta, particularly if you are not sure, or you want to open it up so that it can be alpha, it can be beta, and both of them will react. So that is what is being shown here. That is this another way of representing these Howard perspective formulas, the ring structures, where the configuration, whether alpha or beta, is not shown. It's open. It can be either of them. And they react. If it was a breakdown, yes, you have that enzyme, lactase, that is acting there. And here again, you see the bond which is formed between the lactose and the glucose molecule. You see that's a beta bond over there. So you see there's the lactose molecule is having this galactose molecule in a beta configuration. And then the glucose molecule is in an alpha configuration. And these two, bond, you have that beta 1,4 glycosidic linkage between the two glucose molecule, between the lactose molecule and the glucose molecule to give you, and the galactose molecule to give you lactose. So that is that for lactose. Now, we move on to bigger carbohydrates. See, so far we've been talking about just disaccharides. That is taking two monosaccharides that are binding together, with a glycosidic bond between them, just two of them. But you can have bigger carbohydrate molecules, and these we can classify them into homopolysaccharides and heteropolysaccharides. Carbohydrates that contain the same monosaccharide units. If you didn't understand it when you were in first year or you've forgotten, that is a revision for you. Homopolysaccharide. So you see that what is being depicted here is that you see all of them are the same color, which means it is the same monosaccharide units. So for instance, if it was glucose units, everything, all the monosaccharide units here, they are all glucose. That's what it means. Or heteropolysaccharide, where you have different monosaccharide units. So for instance, if you have like some glucose, and then you have some perhaps a derivative, N-acetylglucosamine or glucosamine, it's a mixture of different monosaccharide units to give you that polysaccharide. That we'll refer to as a heteropolysaccharide. So we represent that with these different colors, to make it clear. Then we can also classify the polysaccharides as either they are branched or unbranched. So you see the branched ones are just straight chain molecules. The bond you find there is 1,4-glycosidic bond throughout. But when you come to branched, you see you have the straight chains, and you also have some branch points. And so you have the alpha 1,4-glycosidic bond, and usually you'll be having 1,6-glycosidic bond as the branch point in these branched polysaccharides. Now we take what we refer to as amylose. So amylose is one of the components of starch and is a homopolysaccharide that is having the alpha 1-glycosidic bond between them. So you see that straight chain as an amylose molecule there. But apart from the alpha 1,4-glycosidic bond that is formed between the glucose molecules, you can also have like a helical structure forming, similar to what you see in proteins. So that's the straight chain, when it folds, you can see there are a lot of hydroxyl groups there. So those hydroxyl groups, and even those the oxygens that are forming the glycosidic bond between the monosaccharide units, can have some hydrogen bonding interaction, which will result into the formation of such a helical structure in amylose. Actually, that part of say starch is what will become a soluble part. And you see amylopectin. Amylopectin is also a component of starch, but this is branched. So you will see that there is that straight chain structure, and then from the straight chain structure, you have some branch points. So you see the branch points on the straight chain, alpha 1,6-glycosidic bond. That is for amylopectin. And the interesting thing here is that you see when you, for instance, you put starch into water, let's say you're forming some aqueous solution or emulsion with such a starch, you will realize that a portion of the starch gets dissolved in water. That portion of the starch which is getting dissolved is the amylose component. Then you also notice that there is an insoluble part of the carbohydrate that doesn't dissolve. That insoluble part we refer to as an amylopectin. So the two of them are in a mixture together, forming the molecule of starch. And you also notice that these bonds, the alpha 1,4-glycosidic bonds, I think whether it's in your class, I mentioned the fact that salivary amylase breaks down these alpha glycosidic bonds, but it's not able to break down beta 1,4-glycosidic bonds, which you find in woody parts of plants. And so that is what we refer to as roughage. So fiber, for instance, is made of, say some cellulose, which we'll also look at its structure as we go ahead. So we saying starch. So starch, which is an energy storage in plants, is either stored in the roots. What you see here is some cassava there. So cassava is that's a root tuber. So the storage of carbohydrates in roots by plants. After the plant undergoes a process of photosynthesis, that's a process of preparing food for itself. And the ingredients it uses to prepare the food are carbon dioxide and water. So that takes you back to the chemical formula, the general chemical formula of carbohydrates. You remember? It was CX, then into bracket H2OX. Remember the name is carbohydrate. So carbon and water. That is where it's coming from. So that a certain number of carbohydrates and then H2O, same number. So we're having CX, H2O into brackets X. Now, you see the plants, they make use of ingredients carbon dioxide and water. And if you look, you have carbon dioxide is containing what? Carbon and oxygen. Water, hydrogen, and oxygen. And so far, as we've been looking at the structures of the monosaccharides, these were molecules consisting of the elements carbon, hydrogen, oxygen. So you see where it's coming from, carbon dioxide and water. Those are the elements that you see in carbohydrates. And they use the energy from the sun to prepare that food, the carbohydrate. And when they prepare it, they store it in the roots, in seeds, in fruits, in the stem, as stem tubers. And we, who are non-photosynthetic, go and take these plants, use them for food to get the carbohydrates from them. Now, we've already mentioned, the difference between amylose and pectin, and the fact that the two of them are present in starch. And the amylose forms the soluble part of the starch, whilst amylopectin forms the insoluble part of the starch molecule. Now, cellulose. So these two, I mean, you think is so much of a complex thing, but really it's not. Just to take a look at it and see that apart from the glycosidic bond, which is forming between the glucose molecules, you also have some hydrogen bonding that is forming, which are shown in dashed lines, with various groups. So you have hydroxyl groups that are forming some hydrogen bond interactions between them, which will keep the molecule, more stable, apart from the glycosidic bond, which is existing in those molecules. And the interesting thing is that the cellulose has the beta-1,4 glycosidic bonds, which saliva amylase is not able to break down. And so, because of that, if you take in, fiber or wood, you will not be able to digest that into glucose units to generate energy. Even though that, compound contains glucose molecules, the cellulose, you cannot break it down. The salivary amylase cannot break it down into glucose units to generate energy. So the alpha amylase is specific. It breaks down only the alpha-1,4 glycosidic bonds. I think I mentioned it, that you see an animal like termites that are eating wood. And it's because you have a symbiotic relationship of the termite with a bacteria, Trachonympha, that is in the hind gut of these termites. So that even though you see that it's a termite that is chewing the wood, itself does not benefit from it because it doesn't make use of it. It doesn't have the enzyme that can break down cellulose. But a bacteria that the termite is in a symbiotic relationship with, the trichonympha, has the enzyme cellulase that can break down the cellulose molecules. So that is why you can see termites chewing wood. Right, now we can take dextrans. Now dextrans have they have a bacterial origin in the sense that bacteria produces these dextrans to form the biofilms, slimy structure, sort of a covering in which the bacteria can freely grow. And the bonds that you see between the monomers in dextrans can be the alpha, you can have the alpha 1,4-glycosidic bond there where you have these glucose units joining to the carbon 4 of the next glucose molecule. But what you notice is that you have the alpha 1,6-glycosidic bond, which we saw as a branch point in cellulose. And even you can have alpha 1,3-glycosidic bond. So that's going to be a bond between carbon number 1, carbon number 3, or carbon number 1, carbon number 6. And these molecules, dextrans, generally are formed by bacteria in the buccal cavity, the mouth, streptococcus, that acts on carbohydrates to form what we refer to as plaques. So plaques actually, have dextrans as a major component. And why you see plaques are so hard, you can see the type of bonds that are between these monosaccharide units are the 1,6-glycosidic bond and then the 1,3-glycosidic bond. So there is more bonding between the monosaccharides. That will make it more difficult for that molecule, dextran, to be broken down. Another usage of dextrans you come across is they are used as lubricants in, say, eye drops, for instance, ear drops. So virtually any medication that comes with that lubricating process in the eye, you see dextrans are used in those molecules. Then dextrins. So pay attention to realize that there is difference between dextrans and dextrins. So you see, for dextrans, we see you have that formation of 1,6-glycosidic bond, 1,4-glycosidic bond, which is much like what you find with dextrins, except that dextrans are formed by these bacteria, from glucose molecules, which is to say, if a lot of times when you were kids, it's advised that you don't take sweets at night, and also to make sure that You don't have carbohydrates, sweet things on your teeth while you go to bed every time. The dentist advises brush your teeth twice a day and things like that. Here, dextrins are oligosaccharides that are produced from the breakdown of glycogen. So the storage form of carbohydrates in the liver or in the skeletal muscles, when they are broken down such that you have some oligosaccharides out of such, because in the glycogen molecule you have branch points. So those branch points will be there. Such smaller molecules from the glycogen molecule is what we are referring to as dextrins. These are used in food processing to enhance the crispness of things like biscuits, the cookies. You see the use of dextrins in these products. It's also used in pharmaceutical products as binders, like tablets. They use dextrins in there as binders. So they bind all the components of a tablet together, for instance. And you will realize that when you buy envelopes, there is a glue on the envelope, but that glue is dry. And when you make the glue moist, then it becomes adhesive, and then you can close the envelope. Now, such glues, contain dextrins. I mean, which is why if you should use your tongue on it, you will see that it has a sweet taste. And that's because of the dextrin. The dextrin is a sugar, and so you have such a sweet taste coming from it, the dextrins. Now, this is just to, show that the glycogen molecule can be that large and have a lot of glucose molecules in it, because it's branched, it's heavily branched, or it's a storage that should have a high surface area. For it to be effective, it couldn't have been just a straight chain of glucose that is being stored as glycogen. If it happens that way, it means at the time that glycogen will be needed to be broken down, it can only break down from one end. Because to be breaking down from only the terminal ends of the molecule, to be giving out glucose molecules into the bloodstream that can undergo metabolism to generate energy. But here you have the glycogen molecule being heavily branched, so that when there is a need for the glycogen to be broken down, there are various ends of this molecule that the glucose molecule should be breaking from. And this is a homopolysaccharide because it's made up of only glucose units. So it's a homopolysaccharide, heavily branched. And you can have up to 50,000 glucose units in a mature glycogen molecule. I mean, if you look, you zoom in much more into the structure of glycogen, you realize the glycogen molecule, if you take the first straight chain as the primer on which there will be the branch points developing, and you take that first branch as tier 1, the branches that come on those will be tier 2, and the ones that come on those branches too will be tier 3. And you can have up to 5 tiers Yeah, no, sorry, you can have up to 12 tiers. 12 tiers. And those 12 tiers, each of the branches will consist of about 12 to 14 glucose units. And so you can see why is that heavily branched and having a lot of glucose units in there, 50,000 glucose units. That's a lot that can carry us through, a period of fasting, like in the nights when we are sleeping and glycogen can be broken down into the bloodstream as glucose, which will then, generate energy to feed us. Now, if you take a look at a glycogen molecule, so you see that's complex structure we were looking at. Now we have zoomed in to see the bonds that are formed within that glycogen molecule. And you notice that there is, apart from the alpha 1,4 glycosidic bonds, which is formed in the straight chain, there is also the alpha 1,6 glycosidic bond. So it's glucose units that are forming straight chain, but at the same time they are branched, and so they have the alpha 1,6 glycosidic bond. So you can see the difference between glycogen and starch. Glycogen is made up of these glucose units, having the 1,4 glycosidic bond and the 1,6 glycosidic bond. If you take starch, you are having it's the same bonds you will see in starch, except that you have amylose and amylopectin, where amylose is a straight chain and amylopectin is not. And so that is the difference. We have chitin. So chitin is a major component in the exoskeleton of insects, of arthropods, of arthropods such as insects, crustaceans. So that will include things like lobsters even as well. You can have chitin also in exoskeleton of things like centipedes, millipedes. And so that's a major component, forming the exoskeleton of insects. Then you can have a derivatization. You can have a derivatization of chitin into chitosan, which we'll come to. But if you zoom into the structure of chitin, you realize chitin is also a homopolysaccharide. It's made up of the same monosaccharide units. This time around, it is a derivative of glucose, and that derivative is N-acetylglucosamine. So you see, we've seen this much earlier, where you see on carbon number 2, there's an amine group on which an acetyl group comes to, bind, conjugate to give you N-acetylglucosamine. And these N-acetylglucosamines, bind, they have a beta linkage between them, and that will form the structure of chitin, which is a major component of the exoskeleton of arthropods. Then I think this should be the last thing. So like I said we should be taking about some 45 minutes. So we are just on course. You have chitosan, being produced from chitin. So you see, we just saw chitin to be a homopolysaccharide, which consisted of N-acetylglucosamine. So you see, the units you see up there, they are all having the N-acetyl group. Because see that amine group that has now conjugated with acetyl group up there. So chitin made up of N-acetylglucosamine. And just deacetylation, that is when you have some of the acetyl groups removed from the molecule so that you have a chitosan. If you look at a chitosan structure, you see the first glucose unit on the right is still having the acetyl group on it, but the second and the third, counting from the right to the left, you see them not having the acetyl group on the amine group. They lose that acetyl group. That is the deacetylation that we were talking about. And so you see that two acetyl groups, this acetyl group up there is removed, it's no longer here. You have an NH2. Here too, this acetyl group is removed. And so this glucose molecule too is having just an amine group. And so in chitosan, you are having N-acetyl glucosamine and glucosamine. So that will be a heteropolysaccharide. That too you find, in crustaceans, in their exoskeleton, like crabs, lobsters. You find that in them. And well, another application in which you find chitosan is that they are used as biopesticide components. It sort of gives some protective coating to plants, for instance, against pests, and is capable of disrupting, cell membranes of some some bacteria that, like plant bacteria, for instance. So you find chitosan is used in some biopesticide, and it's even used in bandages, to reduce bleeding. Sort of, it's because it's also having, some kind of an antibacterial activity as well, and so it's used in these products as well, like bandages, on wounds. All right, so this should bring us to the structure of carbohydrates, and the next time we meet, we then take the metabolism of carbohydrates. So if you have any questions, you have anything that is not clear, you want to ask anything about it, you can either write in the chat or you raise your hand. I'll call your name, and then I will enable your microphone, then you can speak. So the floor is open. Now I see you are 65 people. Still, your number is low, which means some of you are unable to be live.